Hello dear friends, welcome back again to another video from my YouTube channel. Dear friends, in our previous video we discussed about the development of endosperm. Endosperm is the result of triple fusion, whereas the egg cell in the embryo sac fuses with the male gamete and that results into the formation of zygote, and the zygote get developed into the embryo. That is known as embryo development. Embryo development, technically it is called as embryogeny. Embryogeny. Here geny means generation, formation, synthesis, production, development. That is called as embryogeny. Development of embryo is called as embryogeny. As we know, in the angiosperm there are two types of seeds are there. One is dicot seed and another one is monocot seed. In dicot seeds, dicot embryo get developed. In monocot seeds, monocot embryo get developed. In this particular video, we are going to understand about the development of dicot embryo. And by the end of this video, we'll try to understand what is the matured structure of dicot embryo with the help of neat labeled diagram that is very important for board examination, my dear friends. As we have discussed in the previous video and discussed in this video also, embryo get developed by a single cell which is diploid. It is called as a zygote. It is called as zygote, and zygote is always diploid. This zygote formation takes place inside the embryo sac, and we know embryo sac has got two ends, two poles. One is micropylar end and another one is a chalazal end. Let us label the micropylar and chalazal end for our better understanding. This is supposed to be the micropylar end and this is the chalazal end. They are the two ends of an ovule, anatropous ovule. Now, this zygote, it undergo a mitotic division. And this division is a transverse division. That means cell is going to divide on a transverse plane. ಯಾವುದೇ ಒಂದು ಜೀವಕೋಶ ಅಡ್ಡ ಸೀಳ್ಕೊಳ್ತಪ್ಪ ಅಂತಂದ್ರೆ ಅದನ್ನ ಟ್ರಾನ್ಸ್ಫರ್ಸ್ ಡಿವಿಷನ್ ಅಂತ ಹೇಳ್ತಿರ್ತೀವಿ. कोई भी एक सेल अगर आड़ा कट हुआ तो उसको हम क्या बोलेंगे? ट्रांसफर्स डिवीजन. This zygotic cell undergoes a mitotic division, and what kind of mitotic division? It is a transverse mitotic division. As a result of that, two cells are formed. Now, these two cells, they are diploid. Why they are diploid? Because of mitosis. Mitosis is the equational division. Because of mitosis, these two cells, they remain diploid. Let us write the ends here. This is the chalazal end and this is the micropylar end. From one cell, we'll get the two cells. The cell which is towards the chalazal end, it is called as terminal cell. Or it is also called as embryonal cell. The cell which is towards the chalazal end, it is called as terminal cell or embryonal cell. And the cell which is towards the micropylar end, it is called as basal cell. Why it is called as basal cell? It is towards the base. That is why it is called as basal cell, or it is also called as suspensor cell. Why it is called as embryonal cell and why it is called as basal cell is this cell in future, it give rise to the embryo. That is why it is called as embryonal cell. And this basal cell in future, it give rise to the suspensors. That is why it is called as suspensor cell. We'll understand what is this suspensor little later. Now, after this, the terminal cell undergoes mitosis. Similarly, the basal cell also undergo mitosis. But there is a difference between terminal cell mitosis and a basal cell mitosis. What is the difference? Terminal cell undergo mitosis both transversely and longitudinally. Means cell is going to divide on a transverse plane as well as on a longitudinal plane, whereas the basal cell, it divides only by a transverse type of cell division. बेसल सेल सिर्फ आड़ा कट होते जा रहा, whereas टर्मिनल सेल आड़ा और लंबा दोनों भी कट होते जा रहा, ट्रांसवर्स as well as longitudinal. टर्मिनल सेल ಕೇವಲ ಅಡ್ಡ ಕಟ್ ಹಾಕೋತ ಹೊಂಟದೆ, whereas the ಬೇಸಲ್ ಸೆಲ್ ಯಾವಾಗಲೂ ಕೂಡ ಅಡ್ಡಾಗಿ ಕಟ್ ಹಾಕೋತ ಹೊಂಟದೆ. ಟರ್ಮಿನಲ್ ಸೆಲ್ ಅಡ್ಡ ಮತ್ತು ಉದ್ದ ಎರಡೂ ಪ್ಲೇನ್ ಮೇಲೆ ಕಟ್ ಹಾಕೋತ ಹೋಗ್ತಾ ಇದೆ. Now, as a result of that, we'll get 4 cells formed. This is cell 1, cell 2, cell 3 and cell 4. 4 cells are formed. Now, this is because of transverse as well as longitudinal division, whereas the basal cell is going to divide only on a transverse plane. Because of that, we are going to have these cells like this. All these cells are the suspensor cells, but at the base we have a large swollen cell. This entire, it is called as suspensor. Suspensor is formed by the basal cell, whereas this globe-like structure consisting of 4 cells, they are because of the terminal cell. Why it is globe-like? Because the cells are divided on a transverse plane and the cells are also divided on a longitudinal plane. That is why we'll get 4 cells here. This structure, it is called as pro-embryonal quadrant. Why it is called as pro-embryonal quadrant? Pro-embryonal means it is a future embryo. Pro-embryo, future embryo. And why it is called as quadrant? Here we have 4 cells, that is why it is called as quadrant. Now, here, this is the result of the transverse mitotic division in the basal cell. And this last cell, which is swollen, it has given with a particular name. It is called as haustorial cell. The basal cell of the suspensor is called as haustorial cell, whereas a globe-like structure, it is called as pro-embryonal quadrant. Now, this pro-embryonal quadrant in future, it give rise to the 8 cells. 8 cells are formed. From 4 cells, we'll get the 8 cells. We'll have the 8 cells. Let us draw the 4 cells first. They are the 4 cells that we have. It is a pro-embryonal quadrant. Then we'll have the 8 cells formed. 1, 2, 3, 4, 5, 6, 7 and 8. 8 cells are formed. Here the suspensor cell, this suspensor cell, the first suspensor cell which is in contact with the pro-embryonal quadrant, this cell I am talking about, this cell, the cell which is in contact with the pro-embryonal quadrant, this cell, it grow in its size, it become bigger, this cell becomes bigger, this cells becomes bigger like this. Remaining cells, they remain in their respective size. They remain in their respective size. This is the hostorial cell, and all these cells are the suspensor cell, whereas the cell which has increased in its size, it is called as hypophysis. Hypophysis cell. This is the hypophysis cell. It is the suspensor cell only, but the only thing is it has increased in its size. That is why it has given with a particular name, that is hypophysis cell. Now from pro-embryonal quadrant, this entire structure become pro-embryonal octant. Why it is octant? Here we have 8 cells, that is why it is octant. Why it is quadrant? Here we have 4 cells, that is why it is called as quadrant. Let us label this. It is the pro-embryonal octant. It is the pro-embryonal octant. After this, what is going to happen? After this, let us write here the next step. The pro-embryonal octant, which is supposed to be like this, 4 cells, 8 cells. They are all the cells of pro-embryonal octants. We have 8 cells here. Let us draw. Before we draw this, pro-embryonal octant, pro-embryonal octant undergoes periclinal division. Pro-embryonal octant, that means this entire structure, it is called as pro-embryonal octant. Pro-embryonal octant undergoes a particular division, that is called as periclinal division. What is periclinal division? In periclinal division, if one cell divides into 2 cells, out of 2 cells, 1 cell is added inside and the other cell is added outside. That type of cell division is called as periclinal division. Suppose if this cell divides, it results into the formation of 2 cells. 1 cell is added inside and the other cell is added outside. This cell is added outside and this cell is added inside. This type of division is called as periclinal division. Periclinal division is nothing but a cell division in which if one cell divides into 2 daughter cells, out of 2 daughter cells, 1 daughter cell is added inside and the other daughter cell is added outside. All the octant cell, they perform a similar type of periclinal division. As a result of that, we'll get a structure like this. Now, this is the hypophysis. Hypophysis also undergoes transverse division. Hypophysis cell undergo transverse division, and all these divisions are mitotic divisions. There is no meiotic division in the entire embryo development process, my dear friends. Now, this hypophysis, it divides by mitosis and results into the formation of totally 8 cells. 1, 2, 3, 4, 5, 6, 7 and 8. Hypophysis cell divides transversely by forming the 8 cells. Then at the base we'll have this suspensor cells. They are all the suspensor cells. And here we have the haustorial cell. This is the suspensor, and these cells are hypophysis cells, and this is the pro-embryo. At this stage of the embryo development, it is called as globular shape of embryo. Globular embryo. This shape is called as globular embryo. Now, what is going to happen next? What is going to happen next? And the cells which are formed outside, in the pro-embryonal octant, all these cells, they are called as dermatogens. Or simply they are called as epidermis, epidermal cells or dermatogens. Derma means skin. They form a skin-like structure on the pro-embryonal octant. That is why they are called as dermatogens or the epidermal cells. Now, here we'll have 8 cells in the hypophysis. Out of 8 cells, these 4 cells, 1, 2, 3, 4. These 4 cells, they form radical. They form a radical. In future, they form the radical. That is the fate, that is the future of all these 4 cells. 1, 2, 3, 4. The basal hypophyseal cells, they form the radical. These radicals, they include root tip and root cap. Root tip and root cap of the embryo is formed by the 4 cells which are at the base. Now, these 4 cells, 1, 2, 3, 4. Out of 8, the basal 4 cells, they form the radical that includes the root tip and the root cap, whereas the upper 4 cells of the hypophysis, they form the root cortex. Root cortical cells. They form the root cortical cells or they form the root cortex. Whereas, let us erase this, whereas this half of the globe, this much, half of the globe, these cells in future, they form the hypocotyle. They form the hypocotyle of the embryo, whereas the remaining half of these cells, they form the plumule. They form the plumule. They form the plumule and they form the cotyledons. Cotyledons of the embryo. This structure is very, very important for competitive exams, my dear friends. In globular shape of embryo, the basal 4 hypophysis cells, they form the radical of the embryo, whereas the half upper portion of the entire globe shape, they form the plumule of the embryo, and the cotyledon of the embryo, and the base of this much portion, this will give rise to the hypocotyle, and this 4 cells, 1, 2, 3, 4, these 4 cells, they form the root cortex. After this, the cells at this angle, and this angle of the globular embryo, the cells of these two angles, they divide more, and they give rise to a heart shape. Heart shape of the embryo. Here, these two are our future cotyledons, whereas in this depression there is a plumule located. Here, plumule is located at this depression, and why this structure is attained? Because at this angle of the globular shape of embryo, there is more cell division, where at the center there is less cell division. As a result of that, there is a depression appears and there is a more cell division. That is why this angle and this angle of the globular shape of embryo, they grow more as compared to this central part. Now, after this structure of the embryo, it is called as heart shaped embryo. Heart-shaped embryo, and this is the globular embryo. Why it is called as globular embryo? Because it appears like a globe. That is why it is called as globular embryo. Then after this, the structure develops like this. These cotyledons, they further divide the cells which are at this tip and the cells which are at this tip. They further divide and they grow further. As a result of that, we'll get a structure like this, which is supposed to be our embryonal axis, and these two cotyledons, they bend like this. Let us shift this micropylar end here. They bend like this. They bend like this. These two are the two cotyledons. They grow like this, and there is a less space in the embryo sac. Due to the less space, they get bent towards one side. Here, these two are the cotyledons. They are the cotyledons. Whereas this entire stalk-like structure, it is the embryonic axis, and this upper portion, it is called as epicotyle, and this lower portion, it is called as a hypocotyle, and at the base, here we have a strong tissue. This tissue is called as the root cap. Just above the root cap, here we have the root tip. Together, they constitute the radicle of the embryo. Radicle give rise to the root system in the plants, whereas the shoot system is produced by a plumule which is situated here. This is called as plumule. This is the plumule that give rise to the shoot system, and radicle, it give rise to the root system, and there are two cotyledons. And these two cotyledons, they protect the plumule. They protect the plumule during the seed germination. This is the matured structure of dicot embryo. It seems to be very complicated, the entire process, but it is very simple, my dear friends. One cell is getting divided into two cells, and further it undergoes many series of cell division and results into the formation of many kind of structures. From globular, now it becomes the heart shape, and from heart shape this becomes the horseshoe shape. Horseshoe shape of embryo. In majority of the competitive exams, the sequence of embryo development is repeatedly asked, from globular to heart shape, and from heart shape to the horseshoe shape. Horseshoe ಅಂತ ಹೇಳಿ ಎದಕ್ಕೆ ಕರಿತೀವಿ ಇದಕ್ಕೆ ನಾವು? ಕುದುರೆ ಕಾಲಿಗೆ ನಾಲ್ ಬಡ್ದಿರ್ತಾರಲ್ಲ, घोड़े के पैरों में जो नाल मारे रहते ना, ऐसा दिख रहा है, इसलिए it is called as horseshoe shape. This is the entire process of development of dicot embryo. In our next video, we are going to understand the development and the detailed structure of monocot embryo. Dear friends, if you like this video, please hit the like button and please subscribe to my channel for your future videos. Thank you so much.